Hello and welcome to the live lecture today, coming to you from the University of Kent. I'm Dr Alexandra Moores. It's lovely to have you in. We'll wait a couple more minutes to see if anyone else is going to join, and then if not, then we'll get started. So please feel free to pop any questions you have in the chat as you go along, and I'll answer all the questions at the end. So welcome everybody to our Zoom live lecture from the School of Biosciences at the University of Kent. So this is just a quick session to introduce you to some of the team at the University of Kent in the School of Biosciences, and also to give you a bit of a background to some of the topics that you'll be covering and you should have covered during your previous studies through your degree. So life in biosciences. What's it like to study at Kent? Well, I've been at University of Kent for quite a number of years in various different roles, and I've thoroughly enjoyed it. So I'm a lecturer in microbiology, and I teach on a various number of modules throughout each of the stage 2 and stage 3 up to master's level. So I want to start off just by asking you to, when you have time, go and fill in the Student Success and Expectations survey. We want to get to know you, and this will give us a little bit of indication of, what your expectations are, what your concerns are, and what your feelings are about learning at the University of Kent. And this also includes some blended learning, and they'll be explained to you along the way. So where can you find this survey? You can find it in the Get a Head Start at Kent Thinkific site, so the one that you've just come from, where you got the Zoom link from. So you can find it in a section that says Studying at Kent, Your Learning Journey. And at the top you can see there's a little section saying Your Learning Expectations, and that's where you can find this little survey. It's very short. It'll take you probably about less than 5 minutes to complete, and it gives us an indication of how you're going to tackle your first year. Also, it enables us to put some implementations into your first year as well to really allow you to succeed along the way. And that brings me on to the next slide. So study support. So what kind of support are you going to get at the University of Kent, and specifically within the School of Biosciences? Well, we have the Student Learning Advisory Service, and this is a central team across the university that will set up individual and group study skill sessions. So anything that ranges from how to write essays all the way up to presentation skills, anything that you might lack a little bit of confidence in, they will be able to help you on that. Additionally, they have maths and statistics clinics as well. So it helps you just refine those skills that you'll need when you come into a biosciences degree. They've got academic writing skills that'll help you with all of your coursework. So preparing for lab reports, or maybe some essays, or even essays when you come to the exams as well. And they offer a large range of different learning resources. You'll also have an academic advisor. So every student gets one, and they give you one-to-one advice and guidance throughout the entire of your degree. They'll offer you what's called a personal academic support system, or PASS. So they'll look at your grades and how you're getting on, and they'll give you guidance and support along the way. This is usually through progression, monitoring, and reporting. So it just gives you a little bit of help, and also allows you to discuss your progress with them. We use a few different tools for e-learning. So we use Moodle. Maybe you've used Moodle in the past. It's a platform we use to put all of our lectures on, all of our PowerPoint presentations that you'll be using for your lectures, all the notes you need going forward. We also have MyFolio, and this is something that is really useful if utilized. So this will help you plan your career progression along the way. Finally, we've got something really new and exciting coming to the Division of Natural Sciences. This is the Natural Sciences Skills Hub. So this is going to be a resource for you that will support you throughout the entire of your degree, that offers essential skills training specifically for your program of study. So as a quick whistle-stop tour of support study, if you do have any questions about any of the support, please just pop them in the chat and we'll be able to answer you at the end. So let's move on to Welcome to Microorganisms. So this is something I teach throughout stage 2 and stage 3 and into the masters, but you will be working on this from stage 1. So from your first couple of weeks, you'll be learning about microorganisms, and you'll actually get some hands-on experience in the laboratory working with microorganisms along your degree as well. Now this session is tailoring up with the catch-up compendium. All of this is on the Get a Head Start at Kent Thinkific site, the site that you've just used to join this Zoom session. And it features in the biology section under microorganisms. So everything that I present today will be in the catch-up compendium, part of the biology section. But we've also got the chemistry and maths and statistics sections as well for you to have a look at. So if you feel a little bit rusty on some of these areas, please feel free to take a look at your own time going forward before you start. Just to note, it is not essential that you know all of this. We will recap all of this. So please don't worry, don't stress that you're not sure, because you can ask those questions when you do start. So what will we be covering today? The theme today is microorganisms, and this is chapter 7 of the Catch-up Compendium Biology. We'll be looking at bacteria, viruses, fungi, and parasites, and a little bit of background around each of these topics. So shall we get started? So microorganisms are hugely diverse. They are defined by size, because they're extremely small, and they are unicellular in most cases. You'll see in parasites that differs slightly, but we'll go into more details later on. Most of the organisms are harmless, and they often feature as a commensal on the human body or inside the human body. So for example, there's an image you can see that is just a graphical representation of what the human microbiome might look like. So there's a huge diversity of different organisms in the human microbiome. So that's in your stomach and into your digestive system. These play an essential role and often don't cause us as humans any harm to health, really. And often they can be beneficial. So they can cycle nutrients, they can also utilize food products in your stomach to break down various different compounds. We can also use microorganisms in other ways. So we can use them to make beer, wine, bread, all the good things, but also we can use them to make antibiotics and also engineer them to make recombinant proteins as well. So there's a huge diversity of roles of microorganisms in the world. However, some microbes cause disease, and we call these pathogens. So in pathogens, we'll go into a little bit more detail for some examples along the way today, but there are 4 main groups of microorganisms we're going to be covering. So bacteria, viruses, fungi, and parasites. But first, I want to explain to you a little bit why the study of microorganisms is so important. It's because the mortality, so death rate, due to infectious disease is still really high across the world. In the table, you can see a few different diseases caused by different microorganisms, in addition to the number of deaths that was seen in 2019 from these microorganisms themselves. So for example, the first one is tuberculosis, and this is caused by a bacterium, Mycobacterium tuberculosis, And in 2019, it caused 1.4 million deaths. That's nearly on par with how many deaths we've seen from SARS-CoV-2, which is the virus that causes COVID-19, and that was in 2020. So you can still see that actually bacterial species can still have huge mortality worldwide. We've also got HIV, which causes AIDS. This is an RNA virus, and again, that's hugely spread across the world. It killed 0.6 million people in 2019. We also have malaria. This is a disease caused by a parasite called Plasmodium falciparum. Again, it's got a huge number of deaths across the world. And you can see the other examples, again, more viruses and more bacteria. Now, often these cases are seen in low-income countries, and they have a high prevalence of deaths because of a single infectious agent. So that's the organism. Many of these diseases have limited drug therapies. So we find it, as scientists and doctors, really difficult to treat and develop new drugs to treat these organisms. And often they do not have effective vaccines. And that's why they're so prevalent and cause huge mortality. Additionally, those that we can treat with some drugs now have become what is termed multiple drug resistant. So those drugs are having less of an effect on that organism that we're trying to treat the patient for. So it shows you that we still need to keep understanding the biology, the biochemistry of these organisms themselves. So along the 3 years of your degree, you will be understanding, the basics, the backgrounds of each of these diseases, and you'll build up on the knowledge and start to understand them at the molecular level as well, and how it might impact the human immune system. So we're first going to have a look at bacteria in a little more detail. So what are bacteria? Well, they're quite different from plants, animal, and fungi, which are eukaryotes. Bacteria are a different classification. They're termed prokaryotes, and they're usually a single cell organism. They don't have a nucleus, which means that their genome is free within the cytoplasm. And they do not have membrane-bound organelles, which plants, animals, and fungi do. Their DNA, as I just said, is found in the cytoplasm, as you can see in the figure, is free just to interact with all the proteins that will be inside the cytoplasm. And it's usually found as a single circular strand. They also have ribosomes, but they're much smaller in prokaryotes than in eukaryotes. So just as a reminder, ribosomes enable RNA that's been transcribed from DNA to be translated into protein. So that's the central dogma of life. And we'll go into more detail about that when you come into biochemistry in your first couple of weeks. Bacteria also have a very rigid cell wall. So they've got a cell membrane and a cell wall, and then they can be surrounded by something called a capsule. And this can make it really slippery for a white blood cell to grab hold of. Additionally, they have projections, so small hair-like structures you can see on the outside of that bacteria called pili or fimbriae. So these allow the bacteria to attach onto cells. So sometimes they attach onto your urinary tract, and therefore can cause a urinary tract infection. Or they can utilize these small hair-like projections to crawl up cells. They also have longer projections called flagellum. So these allow the bacteria to move, to propel themselves forward so they can move around in the environment. And this usually is in response to a nutrient cell response, or maybe a changing environment condition. Again, you'll come into this in more detail as you go throughout your degree. We're going to have a little look in more detail about the cell wall. So the bacterial cell wall can be separated into two different types, and this is how we classify different types of bacteria. So the cell wall usually consists of peptidoglycan, which are polysaccharide chains that are cross-linked by peptides. And in the image, you can see there are two different types, gram-negative and gram-positive bacteria. Now, gram-negative, which is shown on the left-hand side, have a very thin layer of peptidoglycan. So that's shown in purple. They also have an outer membrane, as well as an inner cytoplasmic membrane. And that's separated by the periplasmic space. The periplasmic space can have proteins in there and can have response proteins that respond to the environmental changes. Also, in both of those membranes, you can have proteins, but also in the outer membranes, you have porins that allow different substrates in and out of the cell. While gram-positive on the right-hand side have a very thick layer of peptidoglycan, and they do not have an outer membrane. And this is really essential, knowing the difference when we come to classify these two different types of bacteria. And I'll show you why. One way in which we can define them is by using a gram stain. Maybe you've done this in the laboratory during your course, your previous studies. If you haven't, this is something that you will be getting expertise in straight away from stage 1 of your degree, so your first year, and you'll be utilizing that in stage 2 as well. So the principle of gram staining is the ability for the bacterial cell wall, which we've just had a look at, to retain crystal violet dye. And I've got a really nice video to show you in a second that you'll actually have an ability to use throughout your degree to understand the principles of a gram stain. So like I said, it differentiates between gram-positive and gram-negative organisms. Why is it important to do gram stain? So it allows us to identify different bacterial species, but also it allows us, because we can define these two species, know which maybe antibiotic treatment you might want to give a patient, for example. So defining the outer membrane is really key. And like I said, you'll be doing this in the laboratory. This is super exciting. But here I'm going to show you a tool that we use to help you and guide you through gram staining even before you get into the laboratory, so that you already have the skills and the understanding of how to do this. And this is a resource called Learning Science. So it's an interactive tool, and I've just recorded me having a go at the tool. So if I press play, we should start to see the video. It'll always give you a bit of an introduction to what you're going to learn. And when we click start, that's when we head on into the instructions. So it'll give you a brief overview of what a gram stain is. And we can zoom in on different functions and have a look at the gram-negative and positive cell walls of bacteria. And again, it gives you some suggestions of what to do. Here we need to rearrange some of the labels. So if we put them into the right place, we know that peptidoglycan is that big layer here, the outer membrane. We've already seen this on the outside. So we pop it here, and then lipopolysaccharides are on the outside. And we get to check if that's right. And if it's right, we get to move on to the next section. So you'll be able to work through tools like this in preparation for your lab skills. So it'll even demonstrate to you how to do a gram stain in real time. So it'll show you different types of dyes that you'll be using and why. And it'll allow you just to work through this gram stain along the way. I'm not going to show you the whole video, but it's a really useful tool for you to be able to go and have a look at when you do start university. So moving on from the gram stain, what you'll end up with when you actually do a gram stain is some cells that are stained depending on their cell wall components. And this is what you'll see under the microscope. So you can see on the left-hand side, you've got some pink cells. So anything that stains pink will be a gram-negative bacteria. And on the two left-hand side, the one in the middle, the central one, and sorry, I got that wrong around. So on the left-hand side, you've got the pink cells. The middle, you've got purple, and on the right, you've got purple as well. Sorry about that. On the purple cells, they are gram-positive. Once you have stained your cells, you can look at these down the microscope. Again, this is what you'll be doing in the laboratory. And you can see here that some cells are rod-like shapes, so long stick shapes, those that are on the left-hand side, and these are called bacilli, while the central image and one on the right-hand side are cocci shape, so circular. And we can go to define these even more. So in cocci, they can be either in long strips, which is shown in the middle, and these bacteria are often called streptococcus. And then on the far left, the far right-hand side, you have some clusters that look like grapes, so look like bunches of grapes, and these are often staphylococcus bacteria. And you'll learn and be able to define all these types of bacteria when you start stage 1. So we know that bacteria also have a slight range in size. They are quite small. So they range between 0.5 and 5 micrometers in length. So that's 1,000 times smaller than an ant. But you can still see them under the optical microscope. So that's the resolution you'll be able to see those at. Again, you'll be doing this in your first year, which is super exciting. You also will be learning about bacterial cell division. So bacteria reproduce by cell division by a process called binary fission. So the DNA will be replicated to give 2 identical copies in one bacteria, which you can see in figure B. And then they will then separate towards the poles of the cell, so each end of the cell, and then there'll be separation in the middle of that one cell to create 2 separate cells. And this is a super rapid process. So this can happen at the minimum of every 10 minutes. This replication can occur in bacteria. In comparison to eukaryotic cells, so human cells, which can take 24 hours to undergo that process. And that's why it's really important in early infection. There's a race between the immune system of the host. So if you're infected, your immune system needs to respond really quickly to this rapid replication of bacterial population before it goes out of control and then will cause disease. So you'll be learning the molecular biology of this process in more detail along the first 2 years. So let's have a little look at an example of a bacterial infection. First of all, we should note that there are about 16 million deaths per year worldwide caused by bacterial infections. That's a significant number of deaths worldwide per year. And this is why it's so crucial really to understand the biology of bacterial infections. There are roughly 100 pathogenic bacterial species. So pathogenic species are those that can cause disease. One example is Neisseria meningitidis. And this is a microorganism, so a bacteria that can cause meningitis. It is a gram-negative Diplococcus bacteria. Diplococcus means it's 2 cells together, which is shown in the image at the bottom right-hand side. So 2 cells that are stuck together. And that bacteria will live in about 20% of the population up their nose asymptomatically. What does asymptomatically mean? It means it does not cause any symptoms. So it will happily live up the nose of that individual, and it won't cause disease. However, if that that person then becomes immunocompromised, or if there's a problem with their health, then it can cause disease. And what can happen is there is an invasion from the oropharynx, so that's inside the nasal cavity, and it will transfer across into the bloodstream. And this is where the trouble starts. So this is when you get septicemia. There's a buildup of bacteria in your bloodstream. And what can happen is that bacteria then can cross the blood-brain barrier, and it can cross into the brain. Now, this is really, really significant. And this is where the bacteria can then cause infection in an area which there isn't enough maintenance or regulation of immune responses. So what will happen is there'll be a big swelling of the brain and the meninges, which encases the brain, which can lead to a variety of different conditions, coma, and in severe cases, death. So you've probably heard about meningitis before, and you can use the glass test, which is shown at the top, that you can push against a rash that's often associated with meningitis infections. And that rash is often caused by a damage to the tissues caused by toxins released from this bacteria, Neisseria meningitidis. So you'll be learning about this in much, much more detail when you come into your second year, and you'll be learning about those type of infections and how to identify which organisms will cause which disease. And also you'll be looking at case studies that actually you have to identify a organism that's causing a specific type of disease as well. We're next going to move on to viruses. So viruses cannot survive independently. So what does that mean? They can only reproduce themselves when they infect a host cell. So in the environment, they'll only be able to survive for a very short length of time before they cannot replicate. So they have to infect a human host, or an animal host, or even a plant host. So viruses can infect animals, plants, but also bacteria. And a bacteria virus is called a bacteriophage, and you can see that on the far right-hand side in the center, in the central column. So they're very diverse, and there's a huge number of different variations of viruses. Again, some cause no health problems at all, while some have significant health consequences, which I think we've seen from the pandemic over the last year and a half. So they use the machinery from host cells, whether that's human, plant, or bacteria, to replicate themselves. So usually they use host ribosomes to translate proteins. They also use sometimes RNA polymerase to copy their DNA. They use to create RNA, and also they use other host machinery to copy their genome. So they're quite sneaky in doing so. And that also allows them to be very small. They won't have a lot of genes encoded on their DNA or RNA genome. So that means that they can really be between 5 to 300 nanometers in their size. And that's 1,000 times smaller than a bacteria. So that's taking you off the scale of being able to look at them down the microscope. So you must look at them under the electron microscope. So much more high-powered magnification. So like I said, they've got DNA, RNA genome, and we can have a little bit look more detail in the virus structure. So here's an envelope virus, but you can also have naked viruses as well. Naked viruses do not have an envelope. They only have the nucleocapsid. So nucleocapsid is a structure that encases the virus genome, whether that's DNA or RNA. If it's an envelope virus, it'll then have proteins that create a very stable structure around the nucleocapsid. And that's often encased with, separated by viral tegument as well. Some viruses also encode envelope proteins that project out of the envelope. For example, SARS-CoV-2, which we'll have a look at in a bit. And these are usually the way in which the bacteria can bind onto the host and enter into the cells. So as I said earlier, some viruses encode genes for replication, but also some rely on host machinery to do this process for them. And we can have a little bit look in more detail about the viral replication or life cycle. So you've got an image on the right-hand side of a virus first attaching onto a host cell. And it can either do this by binding with envelope proteins that will then bind to receptors, so another type of protein on the outside of the host cell. Additionally, they can enter via endocytosis as well. So they can be absorbed into the membrane. Often that's seen in capsule viruses. Once the virus is inside, it'll go through a process called uncoating. So it will release all of its genome into the host cell, and that's when it can either undergo replication or translation of proteins, depending on what kind of virus it is. So this is where it takes over the host cell to start replicating new viral proteins, new viral genome, and then be able to form new viruses. Once that happens, once that new virus has been formed, it can be released either through budding. In this image, you can see budding process, or it can build up so much of the virus in that one cell, that host cell, it can lead to lysis. And that's where there's bursting of that host cell. And that's where that virus is then released into the environment. Or it can be released and infect other cells. So let's have a look at something that's been quite prevalent in the media, SARS-CoV-2. So this is the virus that causes COVID-19 disease. It is a member of the Coronaviridae family, and it is a single-stranded RNA virus. So its genome is RNA, not DNA. It encodes a spike protein, which you've probably heard about, on its envelope. And this is the protein that allows binding to our cells that are found often in our lungs. So we inhale the virus and it binds onto a receptor called ACE2, which stands for angiotensin converting enzyme 2. Don't worry, you don't have to remember that. And this is just to say, this is just additional information. This is not part of stage 2. This is just interesting information for you to go and take home with. So ACE2 receptors are found often in the alveoli or the bronchial epithelial cells that are found in the back of your throat or down into your lungs. So this is where the the virus will bind onto first of all. And this will, once it's bound, it'll enter into your lungs and it'll start the replication process. Now, this virus, SARS-CoV-2, triggers an immune dysregulation in your cells. So, and it causes your cells to overreact using an immune response. And this is what causes damage to your cells. So it's actually host-induced damage. It's what you see as a result in COVID-19. There are a variety of different risk groups. So those that will be infected or lead to more serious disease. Those often individuals have maybe cardiovascular disease, diabetes, or some respiratory issues, or those with an immune deficiency. So those that cannot produce an immune response well enough. Now we know that there have been vaccines developed, and we'll go into this a little bit later on. But if you're interested in something about this, this is, the topics we like to try and keep you as up to date as possible during your studies about what's going on in the world. And this is one of the topics that we'll we will discuss throughout the 3 years of your degree. Next, we move on to fungi. So yeasts, molds, and mushrooms are all classified as fungi. They are eukaryotic organisms, and they can be single cellular up to multicellular. And there's a variety in the kingdom from, and they're slightly separated from plants and animals, but they're still extremely diverse. They can grow as thread-like filaments that you kind of see in the yeast, that forms a really long structure called hyphae. But also they can grow as much more structured organisms, such as mushrooms. They are membrane bound, so they have nuclei and they have cellular organelles as well, and they reproduce either sexually or asexually, or in some cases, both ways. So I'm going to just explain to you one example. Often fungi are seen as opportunistic pathogens. So they cause disease in plants and animals, and often in humans, it's an opportunistic infection. So that means it takes advantage of those with a weakened immune system. So those without a good enough immune system may react differently to those that don't when you have an infection with a fungus. So for example, skin infections, you can see ringworm, athlete's foot, or thrush. And under normal circumstances, that can be an irritant. You have got an infection, but it won't cause serious disease. However, in immune compromised, so those with a weakened immune system, if they get infected by an organism that causes these kind of infections, that can lead to more serious consequences. Additionally, there can be more, fungi can release spores. So these are small particles that can be inhaled into the lungs. Again, if you have a weakened immune system, this can lead to serious disease. And one example of this is Aspergillus. Finally, we're going to move on to parasites. So this is a large, diverse group of infectious agents. They can be single-celled or multiple cellular. And you can see just from the image here, they can be a variety of shapes, sizes as well. They have a very complex life cycle, and they can live in loads of different states in the host at different times, which makes them really difficult to treat. There are 3 main subgroups that we cover across your degree. Protozoa, such as Plasmodium falciparum, which we'll come into more detail in a bit. Helminths, which are worms, and arthropods, which are fleas, ticks, and spiders. So protozoa are the smaller of the 3. They are single cells, and they are about 1 micrometer to several millimeters in length, so slightly bigger than a bacteria. They're generally free living in the environment, and they can also live intracellularly and extracellularly as infectious agents. So intracellularly, they can live in your blood cells, actually in the blood cell itself, and they can also live in brain tissue as well. And they often obtain their nutrients directly from that host cell. While extracellular live in the bloodstream or maybe in the gastrointestinal tract, and they gain their nutrients often by ingesting host cells. That's really grim, isn't it? And one really key example that really does cause a lot of deaths globally each year is malaria. And this is caused by a protozoa called Plasmodium falciparum. Now, often we think of malaria being caused by mosquitoes. That's a bit of a misconception. The mosquito actually transfers that parasite from itself into the human bloodstream. So it's a vector. It enables it's a carrier of that parasite. And that parasite is called Plasmodium falciparum, which you can see on the right-hand side. So it can change shape throughout its life cycle. And it is often found in female mosquitoes' saliva. So when a mosquito bites you, it will inject some of its saliva into your dermal tissue, so into your cells. And doing so, it will transfer the Plasmodium falciparum, that parasite, into your cells, into your dermal tissue. Those parasites can then lead and drive into the bloodstream. They're transferred to the liver, where they replicate to thousands and thousands of more parasites, and then they can enter into red blood cells. And then they go through a continual reproduction cycle in the bloodstream and the blood cells themselves. It leads to anemia and all sorts of really terrible side effects for those that are infected with this parasite. Again, you'll be coming into much more detail into stage 2 and stage 3 of your degree. Next, I want to talk about helminths. So these are parasitic worms, and they're multicellular. They can be extremely small, but can grow into meters of length. And I'm sure you've probably seen videos of individuals that have tapeworm or roundworm infections inside their gut. But here's an image on the right-hand side of a hookworm that is literally attaching and biting into the human intestine. There are 3 groups. There are tapeworms, flukes, and roundworms, and you'll cover them in a lot of detail when you come into stage 2 and 3. But they're easily transmitted to humans, either through swallowing of the eggs or larvae directly, or you can actually swallow them through ingested tissue. So if you had an undercooked pork steak, sometimes the eggs can be transferred that way, and that can give you tapeworm. They can also penetrate your skin. So hookworm, as you can see on the right hand side, that can enter through your feet. So if you stand on a larvae stage of hookworm, it can catch on and it'll burrow into your skin, and then it'll transfer all the way into your tissues. But they can also be transferred by a bite of a vector. So for example, a fly. Finally, we have arthropods. And these are invertebrate animals, such as insects or arachnids. So these cover lice, fleas, mosquitoes, mixed ticks, and spiders. And they impact human health in 3 different ways. So they can be a carrier of other infectious agents, just like we saw with malaria. So we know that the mosquito carries that parasitic infection, which then can be transferred to you. But they can also be parasites within their own themselves. So they can be, just like the hookworm that burrows into your feet, you can have lice that can burrow into your scalp. And finally, they can cause serious allergic reactions as well. You've probably heard of dust mite. Quite a few people are allergic to dust mite. So that was a real whistle-stop tour of infectious agents. So how do we treat them? So treatment relies often on drugs or compounds that attack the pathogen primarily through their own pathways. So pathways that are not shared between the eukaryote and the prokaryote. So drugs will often target prokaryotic systems. And they shouldn't harm the host themselves. So it shouldn't harm you. Now, there are different types of antimicrobials. And I love this image on the right-hand side because people often get confused about what antibiotics do. Antibiotics are for bacteria, antivirals are for viruses, antifungals are for fungi, and antiparasitics are for parasites. And we often hear, and it's always a bit of a joke, when somebody says they need antibiotics for their cold. The common cold is caused by a virus, so therefore antibiotics would have literally no effect on a cold. So it's a bit of a joke, really. And you can see that Batman's telling Robin off there. as I mentioned earlier, with any selective pressure, there comes resistance. So you've probably heard of antimicrobial resistance, and this is where the organism, whether that's a bacterial virus, parasite, or fungi, will become resistant to the drug that you're using to try and treat them. And this is on the rise. It's something that's really, really important in human health at the moment, and is really quite worrying going forward. And you'll get to learn about this and what pathways the organism can change to adapt to drug treatments. So you'll learn about that in much more molecular detail. And finally, before we run out of time, we look at trying to prevent infectious diseases as well. The most common way of trying to prevent infectious disease is through vaccines. So this provides a vaccinated individual with active acquired immunity to a specific disease. So what does that mean? It primes the immune system by either generating an immune response to a specific part of that organism, or by a process on that organism. So it'll allow the body to recognize, target, and then destroy any potential threat, such as a pathogen, in the future. So I've got an example on the right-hand side of how COVID-19 vaccines are working. There are loads of different types of vaccines. You can have viral vectors that encase. So this is a harmless virus that encases a genetic component or a protein component of the SARS-CoV-2 virus that the immune system will then respond to. You can also have the whole virus. So it'll either have a weakened version of that virus or an attenuated form. So it won't be able to cause disease, but it'll look very similar to the original virus shape. So the immune system will respond and recognize that and be able to cause an immune response stimulation when you see the virus again in the future. There's also nucleic acid vaccines as well. And again, we'll go through all of these different types throughout your degree. And these are what's called a prophylactic treatment. So this prevents future infection. But prophylaxis can also be undertaken by also giving low levels of antibiotics as well, or any kind of antimicrobial to prevent future infection. So that was a real quick whistle-stop tour, because we are running out of time today, of infectious diseases, microorganisms, treatments, and also vaccines. So if you do have any questions, please pop them in the chat. I'm happy to answer them in a second. Absolutely fine if not. I hope you've enjoyed today's quick lecture on microorganisms, and during your degree that will be split across many different weeks, and you're going into much more detail and having a look at excitement different infections along the way. And just a reminder for next week, Dr. Emma Hargreaves, another lecturer in microbiology, next Wednesday on the 4th of August will be taking a session on the nervous system. So she'll be explaining how the body survives and able to respond to internal and external stimuli using nerve cells to transmit electrical signals. And she'll cover the principles of the nervous communications and explore the consequences when that signaling goes wrong. So I'd like to thank you for being in today. It's been great having you in, and I'm just going to stop sharing so I can see if you've popped any questions in the chat. Okay, I've got a thank you for today. Thank you very much for listening. I hope you enjoyed it. Please do, if you have time, go and fill in that survey. It would be excellent to hear your views of how you found your learning over the last year and what you want and how you want to learn going forward this year. there are not any other questions, I'm going to sign off and say thank you. I'm Dr. Alexandra Moores, and I really, really look forward to seeing you when you start next year. Take care. Bye.